In this video, we're going to explore the details of hemoglobin, the oxygen-binding protein that red blood cells use to carry oxygen between the lungs and the cells within the body, and we're going to look at the nuances about how hemoglobin knows, so to speak, when to pick up oxygen and when to release oxygen. So hemoglobin is actually made up of 4 individual proteins that combine together, and each individual protein can hold 1 oxygen molecule, meaning that 1 unit of hemoglobin can hold 4 individual oxygen molecules, which actually bind to the iron atom that is found at the center of each of the prosthetic groups called heme. So meaning that because each hemoglobin has 4 iron atoms, this means that 4 oxygen molecules can bind, and collectively these make up the active sites of hemoglobin. Now, hemoglobin has a rather interesting way of functioning compared to other proteins in that the binding of oxygen is actually cooperative between each of the active sites. For example, if we take this oxygen molecule here, even though we see that the active site is relatively closed, 1 oxygen molecule is able to get to the iron and attach to the iron like so. Now, when this first oxygen binds, it causes a change in the shape of the protein, which is what we call a conformational change, and this results in the other active sites of the hemoglobin protein actually opening up. So we can see here that a second oxygen molecule is now able to get to the second iron atom because the active sites have opened up even more, allowing easier access for oxygen. And then when the third oxygen molecule binds to the third iron found on hemoglobin, it opens up the active sites even more, allowing the final oxygen to bind much more easily than any of the previous oxygens were to bind. Now, this kind of answers the question about how oxygen is able to attach to hemoglobin, because as long as oxygen is present, oxygen makes the binding of other oxygen molecules easier to do, but this doesn't exactly answer the question about how hemoglobin knows how to pick up oxygen in the lungs and how to release oxygen when it gets to cells that actually use oxygen. Now, in order to answer why this is the case, we need to look at the optimal temperature and the optimal pH of hemoglobin, because these conditions are actually going to affect the conformation, that being the shape and the openness of the active sites of hemoglobin, which actually allows it to change between the 2 conformations that exist that allow easier binding of oxygen in one case and the complete release of oxygen in the other case. So, this is what we call the open conformation, or what we can call the oxygen binding form. The oxygen binding form, the form that we previously saw that binds oxygen molecules cooperatively, occurs in environments that have a relatively neutral pH, meaning relatively close to 7, although if the pH is slightly basic, so a pH of between 7 to 8, this basically works as well, and this conformation also occurs where the air temperature is lower than that of body temperature, and we call this the open conformation or the oxygen binding conformation, because this makes it easier for hemoglobin to attach the first oxygen molecule, and then cooperative binding ensures that all of the active sites will be filled with oxygen. Now, the opposite to this is the closed conformation, in which all of the active sites basically slam shut, thereby kicking oxygen out of the active site of each of the binding locations of hemoglobin, and this occurs under the opposite conditions. For example, this occurs when the pH is actually slightly acidic, and this occurs in locations where the temperature is at body temperature or higher, and we'll explain why this is suitable for hemoglobin letting go of oxygen when we take a look at where these environments occur. So in this environment, hemoglobin is actually going to release oxygen. Now, the environment that has a relatively neutral pH, or a pH at least, that is close to 7, with temperature that on average is lower than body temperature, is actually inside the lungs. Now, because human body temperature, let's make a note of that here, because human body temperature is approximately 37 degrees Celsius, which is actually quite high. I personally would not want to stay outside for very long if this was the atmospheric temperature. This means that air in the atmosphere usually has a lower temperature than that of the body. We kind of define atmospheric temperature as being approximately 25 degrees Celsius, and that's kind of an average across the entire year. But this is more than 10 degrees cooler than body temperature is, and generally has a pH inside of the lungs of approximately 7, although it might be slightly basic in the lungs due to some residual base. Now, these are basically the ideal conditions for hemoglobin to receive oxygen from the inside of the alveoli when oxygen diffuses to attach to hemoglobin, and therefore this is where hemoglobin is going to bind oxygen and where the cooperative binding mechanism actually functions most ideally. But when we compare these conditions to cells that actually require oxygen, we see that the conditions are quite different, because all cells require energy to be produced, meaning that they are doing cellular respiration. And if we take a little bit of a review about what cellular respiration actually is, this is the chemical reaction where your cells take glucose, which is the most common sugar in the body, and react it with oxygen, breaking it down into carbon dioxide and water, but most importantly, liberating the energy that is stored within the sugar molecule in order to actually produce energy for the cell. Now, because no energy transfer is 100% efficient, some of the energy within this sugar molecule, as it gets broken down, is going to be lost by the reaction, and instead of converting it to a usable form for each of the cells, a lot of this is going to be released as heat. And what this heat is obviously going to do is it is going to increase the temperature of the cells around it, meaning that these cells, the more cellular respiration they do, meaning the more oxygen they consume in order to generate energy for the cell, the more heat is going to be released by these cells, and this is going to cause the temperature to increase a little bit higher than 37 degrees Celsius, providing the ideal conditions for hemoglobin to let go of oxygen and release it into the cells that require it. Now, the other explanation about why hemoglobin lets go at this time is actually because of carbon dioxide, because a lot of this carbon dioxide that is produced by the cells in your body, especially those that require a lot of energy, a lot of that CO2 is going to end up in the liquid part of your blood rather than in your red blood cells, where it can react with water. And if we do a fairly simple acid synthesis reaction, we can see that hydrogen, or that water and carbon dioxide, are going to synthesize carbonic acid together. Now, because this chemical is an acid, therefore the pH is going to drop and become more acidic, providing the ideal conditions once again for hemoglobin to revert to its closed form, to basically shut all the active sites and release oxygen into the cells, where that oxygen can be used to continue the process of cellular respiration. And therefore the conditions ideally in the cells of your body allow hemoglobin to release its oxygen around cells that actually require them. In the next video, we're going to look at some of the drawbacks of this system and look at when this system actually goes a little bit overboard and how the medulla oblongata, the control center for breathing, and the heart regulates the pH of the blood and prevents the excess heat and excess acid from causing problems for the cells around the blood capillaries.